the first session we looked at the upper respiratory tract. Let's move further downwind to the lower airway. So this is the lower respiratory tract. Also called the lower airway or the lower air space. So this cartoon that you have here, this is the trachea. So this bifurcates to form the primary bronchi. Nothing else is visible on this screen, but the primary bronchi will again bifurcate to form the secondary bronchi, and then they bifurcate to form the tertiary bronchi. That I think has been leveled here. And then eventually it goes to the bronchioles till the 23rd generation. Now, what you see on this one is a zoom out from the very distal end of the lower airway channels, like the gas exchange takes place. So these are the pulmonary, this red and blue are the pulmonary arteries and veins, and they extend the gas with the alveolar sacs. So let me write down some comments, part of which I already mentioned in the first session. So we have two lungs, they are shaped sort of conical. So the two cone-shaped lungs are located in the pleural cavities of the thorax. The thorax is a region anywhere under our neck till our abdomen. So the pleural cavity contains this set of lungs on either side of the heart. Functionally, that is important, right? Because the heart is the main engine in our body. It needs the oxygen, and the oxygen is coming from the airway. So it's close over there. Now, I think a couple of weeks back, we were wondering about the structure of the human anatomy, and this is sort of heading into the open question direction. So we have lungs here. Heart is here. We are drawing air into our body. So this is not really the central part of our body. Right? The central part is sort of here in your tongue. So the heart being the engine could have been at the center. Why is it not? What functional benefit do we derive by this structure, all this very critical structure going over here? Because if the heart is centrally placed, it is easier for heart to send blood pumping wise, here it is, right? So the pumping efficiency would be better. It has to spend less energy to send blood to the rest of the body if it is centrally placed. Why is it so high up in the body?
Maybe because there are too many fine veins near the lungs. So it is
why are the lungs there? The lungs could have been
because of pressure.
the pressure drop. Can you explain more? What do you mean by pressure?
Usually,
Pressure of what?
pressure of blood.
By that logic, why don't we have it at the center again? Then it can access the foot and the vein equidistant away. And most of us are short here, but think about someone who is over 6 feet. There the heart is so much high that has to pump blood so far away. I don't have an easy answer for you, so don't expect that I have an answer, but something to think about. Now, one rationale that I came up with is it's a combination of two mechanisms that's going on, right? The lower the lungs are, it would be more difficult to breathe because you have to impose the pressure drop, but this is not the blood pressure. This is the air pressure drop that we have to impose. Just like if you are drinking from a glass, if you're using a straw, it is easier to have a straw which is smaller and you are pulling the glass like this, rather than having a straw that is long, the glass is on the floor, right? So having a short weight pipe makes sense because then you apply less pressure drop. But then comes the question of heart. So it's optimizing between the two. And really I don't have any clear answer. But this itself could be a very interesting bio-inspired mechanics problem. Like why is it like this? It is like this, we know that, but why is it like this? And if yes,
If it is water, the pump has to do more work to pump the blood on the upper part of the mouth.
but it has to do less work to send blood down there.
Maybe in the lower part it can go into some place
So
of
gravity, you mean?
Yeah.
That's a good point.
And if it is located here, then to send the blood from upper part.
But then think about pigs. The pigs are not upright human beings. For them, also it is sort of like on the upper part of the verticality.
Same thing that maybe I didn't rephrase properly because there are too many veins inside this lung area.
Yeah.
So if it is closer to the lung is whatever the reason for the lung is, but if it is closer, then means there is less space to cover in this.
So you are
area.
saying that it is driven by the location of the lung.
Yes, because the concentration of these veins is very
Yeah, that is
high.
true.
Very high.
The gas section has to happen with the lungs. So the vein concentration, the artery concentration is high over there, but your rationale is the heart should be as close to the lungs as possible. So it is near at least.
Independent of the fact why lung is there, I am just telling why it is close to lung.
The lungs. But still, it is a combination of two factors. Right? We can place the lung somewhere else, and then the heart should be there. I mean, what was stopping God from putting the lungs
Because
back over
it
here?
is easy to mix oxygen with blood. So why it is close to the lungs?
Yeah, that is true. That we understand. That it has to be close to the lungs because the oxygen exchange, the alveolar sac takes the carbon dioxide out from the circulating blood. It puts the oxygen in. So it has to be close to the lungs. But why the lungs are there? One argument is, of course, the pressure drop. We have to apply more pressure drop, which is further down, of course. Right? But then why do we have our nose over here and not over here? So these are interesting questions that we don't think about. We just say,
I think it is related to brain.
related to brain in what way?
To transfer blood to the oxygen to the brain.
So you're saying that the respiratory system should be close to the brain?
To easily transport blood to increase the heart beat.
So you're saying that if we think of this as the human body, the heart should be closer to the brain because brain is the main thing that's driving the rest of the
That is the higher or tall and higher risk of heart rate. Then
Those are anecdotal. I don't think it could be proven that way.
So gas exchange, one main thing is need to have large surface area somewhere in the body.
Yeah.
For us, it is this place because the surface area is very high. For fishes, or one thing you need is a big opening where you can get a lot of air because density is low. For fish, it is gills.
Yeah.
For us, it is mouth. So it is sort of optimizing one way that we are eating also from here, and there is this epiglottis also. So one opening is required. Actually, maybe there are too many factors. Too many factors.
Maybe the gravity factor is also important. Gravity is important, but if you think about whole-like animals, then the gravity is distributed out. What I'm trying to imply here is, as I said, I don't have a clear answer, and all your answers are valid answers. But bio-inspired robotics is an upcoming thing. And these are some questions that people have not explored yet. Like, why should the brain be why should the heart be where it is? Why should the lungs be where it is? How does it relate to the location of the brain? And why do we have our nose over here and not over here? Central part of the body. Anyway, so let's move back to the human body as we know it. So the two cone-shaped lungs are located in the pleural cavities of the thorax on either side of the heart. The lungs have some clear anatomical zones that you can sort of think about. and the right lung is not symmetric to the left lung. So we have some level of asymmetry as well. So the lungs have clear anatomical divisions called lobes. And the right lung has 3 lobes. The superior, which is the clinical term for upper, middle, and the lower or the inferior. And then the left lung has 2, sorry, this should be 2 lobes, not 2 lungs. So 3 lobes in the right lung and 2 lobes in the left lung, just superior and inferior. Okay, so as a result, because of the 2 lobes in the left lung, this is also slightly smaller than the right lung. Okay, so now what passes to the lungs, this air that is coming in through the windpipe from the trachea, and then it goes into all these channels. So let me write down the details of that, and some of it we have also covered yesterday when we were talking about the lung generation. So the trachea sometimes also called the windpipe, taking in air from the larynx leads to the conducting airways. As I said, if you just think about the regions of the flux, the air flux passes through in the lungs, conducting, transiting, and then where the respiratory gas exchange takes place. So this leads into the conducting airways. So if I try to draw a very crude sketch, so this is the trachea. So it bifurcates. So this first bifurcation is called the primary bronchi, and then they in turn bifurcate to form the secondary bronchi, and then it follows in a fractal nature, so they bifurcate as well to form the tertiary bronchi. So this is basically the conducting region of the lungs, and if I am so the model that we are developing right now includes still this tertiary bronchi, and what I am assuming in my work, if you are trying to track what is moving to the deep lungs, by deep lungs I mean generation center further below. At this point, with our model, because we are modeling from the upper respiratory tract, which has strong time varying component, it is unsteady in nature, and the flow is going to be unsteady over here. So we end our model here. And whatever is escaping through this so-called outlets, we assume that that is moving to the deep lungs. I don't know a nice way to combine both unsteady modeling and laminar and unsteady turbulent modeling and laminar steady modeling, which is going to happen in the smaller molecules. So I end my model over here and then track what is moving in through these outlets towards the deep lungs. So the nature of the numerical scheme for these two regions of the airway should be ideally different, and I don't know of any work that has managed to merge the two together. Now, from here, Let me write down the definitions first. So the tertiary bronchi would subdivide into several generations of smaller bronchioles. And this, as I said, are the conducting areas, and then they transit into the respiratory gas exchange zone, where you have the bronchioles, the alveolar ducts, and the alveolar sacs. So, this conducting airways then transit into the respiratory zone, zone of the lungs comprising the respiratory bronchioles, the alveolar ducts, and the alveoli. Okay. Now, in terms of the current state of modeling work for lower airway where things become laminar steady, anyone here has seen any work recently? Any paper that you might have read here? So, there's a group at IIT Madras, Dr. Shamuddin knows about them, Mahesh Machakra, they are doing a lot of work for this region beyond. Basically, they do start their model from the lower part of the trachea, but essentially they're modeling everything as steady state laminar typically. And they develop this model which tries to mimic the total surface area of the channels, but it is called a trumpet model. So that's a very useful technique to get some idea about what the flux would be like. But to my knowledge, the details of the anatomy in any kind of modeling work, I have not seen any paper yet. But I would still encourage that you guys look through the papers by this. I think that's true. Now, over here, as you are pointing out the surface area of the gaseous section, that is very crucial. So, you want to guess how big the surface area is and maybe how many times the surface area could be compared to the outer surface area of our body? It is 71 times. So, whatever the outer surface area is, the surface area over which the gas exchange takes place with the pulmonary channels is 75 times of that outer surface area. So the thin-walled sacs called the alveoli are the respiratory surfaces of the lung for gas exchange, and the area is based on the individual, it is between 50 to 140 meters square. So, and the gaseous exchange takes place with the pulmonary channels. And this is approximately 75 times the body surface area. So that's tremendous, and not much work has been done in that. So, and then people have done some work where they are trying to get a reduced model. So there is a group in Israel led by, I think, yeah, so Josu Snistman. I have shared that paper on our share folder. So So there they're looking at, they have developed this CAD models with enough dimensions of the lung, which are essentially planar models, and they're running their flow through those channels, a microfluidic set of channels, and then they're letting some particles in, particles of a suitable size that can pass through those regions. And I think yesterday we were discussing about the internal circulation, but they are seeing that as well in those particulates. And that according to them can have some relevance for the gas extent. But a truly biological model that we can operate using our mechanics platforms, analysis, simulations, whatever, is still sort of an open area. Okay, so this is all that I wanted to write about the lower respiratory tract. I have a very simple example problem for you guys before we move to the next part. So this is, I think, the first figure from a paper that I recently wrote. It has not been published yet. You can find it on archive. So here we are trying to uh, develop an understanding of secondary infection in the lungs. See, the argument is this is the upper respiratory tract, right? The upper airway, that's where the infections primarily start out. Initially, the infection hotspot is over here at the nasopharynx. But after that initial infection onset, doctors have seen that the bronchial infection also emerges pretty soon. Now that initially people thought that that is happening through tissue level replication of the infected cells and as a result the viruses. But the time duration needed for that level of progression of the disease was not agreeing with what they saw in real subjects. And many of this work came from the COVID-19 times. They saw that after the emergence of initial symptoms in the upper respiratory tract, they were developing bronchial infection within 2 to 3 days. And that is not enough time for the infection to progress through tissue level replication to the lower airway. So it was an open question, and I have been discussing about this question with a number of groups, including the IT members group as well. But there was this line of studies that happened in the late 90s and the early 2000s. They saw that this mucus layer that we have, it is viscoelastic in nature. It does undergo some viscoelastic separation. So if you let air flux brush past that mucociliary layer, there could be some particles that can separate out through stretching, and then through separation it can move into and be airborne within the airspace. Now, we also know that if you think about the mucus layers, this is the region where the pathogen concentration initially after the emergence of infections is the highest. What if particles generate from that region get airborne in the airspace, and then we are inhaling them further downwind into the lungs? That way, the pathogens from the upper tract can move quite fast into the lower airway and can explain the secondary bronchial infection. So that was the motivation of this study, and yes, from the work that we have done, it seems like that would be the case. That would be a viable route where particles generated from the upper respiratory tract can move downwind, and because they are moving from here downwind directly, even particulates that are as big as 15 or 20 microns can move directly into the lungs. Unlike 15-20 micron particles from outside, they have to navigate this very complex anatomy, and they cannot, because of the inertial dominance in the mechanics, they cannot move all the way to the lower airway. They will fall out of the streamlines, they will be in the upper part of the nose. But these particles, they get a fairly straight path to follow, and then they move into the lungs. And those being big in the sense 15-20 microns are the biggest particles that we are talking about here. They carry more pathogens, and that can explain the secondary infection. So that was the story in the paper. It is still getting reviewed, and it has been 7-8 months now. It went through the first layer of review, so sometimes things take a while. But the problem that I have is, it's quite fun that I have for you guys, because in the paper I mentioned what the lung generation is in this model, and now I realize that whatever I have in the current draft is inaccurate. So I said that it is still generation 2. Do you agree? Is it till generation 2, this particular picture? As I said, it's already inaccurate, so it's not generation 2. So what is the generation number here? How many outlets are here? 1, 2, 3, 4, 5, 6, 7, 8. So 3, 2 to the power of 3 is 28 in the range of yesterday. So it's the third generation. The funny thing is, before I submitted the paper, we have a respiratory care program in the nursing program in our university. Our respiratory care specialist went through the text in the paper. He said it's all fine. It's in generation 2. So I can actually, I have acknowledged her, so I can blame her as well. So, anyways, I thought I should include this into the material because even I mistook this 6 months back. So, let's write down some comments here on that. Okay, so the above graphic is from, let me write myself in third person here, because if this material is shared, at least someone would know to look up the papers. It's from Dr. Basu's recent paper. Find the bronchial generation number considered in the idealized geometry. So we have 1, 2, 3, 4, 5, 6, 7, 8. Therefore, generation n will have 2 to the power of n outlets owing to the fractal nature of tube bifurcation. Therefore, here the number of outlets is 8. So it is generation 3. So I have to remember to correct this on the manuscript by the time the paper comes back. Okay, so any questions until now? Yes.
I think maybe it can be written in two ways.
Yeah.
It's the end of the third generation, the last point, or the beginning of the fourth generation.
Yeah. Beginning of the fourth generation.
That will remove the uncertainty maybe in reporting sometimes. That we did till the end of the third generation. Because the top part can be the end of second generation.
Right. But typically, if you look at situations, it don't do it that way. Like the non-mechanics community is very well established. It is older than the upper respiratory tract community, and they just refer to the generation number and they're supposed to understand what they mean. So they don't really. What you are suggesting is helpful in the sense that you are spatially locating what the extents of the model are. Like this before the generation post which is useful. Perhaps in our engineering papers we should take that approach.
The result at the end it can be used to maybe study the lower part.
Yeah, that's what I'm doing. So that's what I'm doing. That whatever is coming out here is moving into the new class.
So what is like typical reviews, doctors or engineers doing biology?
So this paper is getting reviewed after 5 like one of the phase channels that we know, like one of the top three. So they are probably going to be visible like us with some biointerest. And this paper does not include any doctors as a co-author. So typically, if you the clinical journals typically would expect some doctors on the co-author list, and also you should be studying something which is more directed towards a particular pathogen or a particular. This is more about a mechanistic study about what is going on. Okay, so let's go to this table. So this is from one of the textbooks that I mentioned yesterday. I think it is from the biofluid mechanics book by P. Nithya Rasu. So I thought that I should include this into the material because it's useful. Take a quick look at what's going on. Initially I got confused with, so for example in generation 16, the table says that the number of channels is 6 times 10 to the power of 4. And I got confused because it should be 2 to the power of 16, right? But if you do a quick check on your calculator, basically it is just the order. So I think 2 to the power of 16 is 6, 5 dot 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 something, and this is 6 0 0 0. So if it's just mentioning the order, so nothing changes based on that fractal calculation. But what is interesting is here, which even I had some lack in understanding on what kind of particular sizes could be entering this channel. So let's go to the last generation 23. So there the size is what? 0.05 microns. So sorry, 0.05 centimeters. So that is around 500 microns. That is wide enough to let all the big particulates that we are talking about in. So we cannot argue that a 20 micron particulate is too big to enter that domain. They can if they are allowed to. It's just that the 20 micron, 30 micron particulates that we are inhaling from outside, because of the inertial dominance in the mechanics, they cannot navigate this band and they deposit in the anterior parts of the nose. But if you have a 20 micron particle generated here, they have a direct access to the lungs. And then they go into the lungs and they have to tolerate those bifurcations, true, but there is nothing stopping them from getting in there. So that size, the typical cross-sectional size was an eye-opener for me. So I had not modeled anything in that domain, in that kind of generation number, but if you guys do at some point, or even if I do, we need to be careful about that. That mechanistically we stay realistic on what would happen and not ignore things. Any questions? Okay.
Yes. Number is it fixed or there are some variations?
The 23? To my knowledge, it is an approximation. And it is not a recent work. It is coming from quite a few decades back. But if you look at a replication of a real one, it is so complicated. I don't think people are that accomplished yet to get a more design. Think about how many numbers are there. 2 to the power 23. You guys have calculator with you? Try that, I'm curious. Can you even generate 2 to the power 23? I know my phone cannot unless I do 2 times 2 times 2 times 2.
Can you? 256 into 256 is already 2 to 60.
Yeah, 8, yeah, and now think about how tiny the ones are. Fit everything over there. And these are 0.05 millimeters, so it's still some width.
Because this fascination of this fractal ordering thing with Mandelbrot brought and all those people, they try to find what fractal is in everything. So, like,
Yeah, and have you guys read about self-organized criticality? There also it's the fractal nature. So there was a generation in the 70s, 80s, they did everything with fractals. Okay, so I wanted to cover this anatomical details because that's where we are heading into. Now we are going to connect the mathematics that we had developed yesterday with a real human respiratory system. So before I move on, any final questions on this? Okay. So,
Just
yes.
question. When you smoke,
Yeah.
how far up is like one smoke particles?
You need to ask when you smoke. I don't know.
I don't want to be personal. So, like, how far it reaches in this order of 2 to the power what scale?
I don't know and I don't want to know.
Actually, I did some sort of this work in my master's, and my supervisor used to smoke. So he asked this question, and I found out.
Okay.
So what I understood, means not an expert, but as I already told you, there was a size thing. That this much size will go here, this much size will go here, and the smaller size will reach to the end.
Yeah.
So as per his request for me to investigate, I found out that it doesn't matter if it's smoke or anything else. It, it has to be of that much size. If there is construction happening and that size is there,
Right.
it will go there, and the silicosis and all those things can happen.
Yeah.
But the point is, if you continuously smoke and some part of this 100-meter square is getting damaged, it cannot recover. The first thing is that it will never recover. Only what you can do is you can try to increase the efficiency of the remaining area using enhanced this blood circulation.
Okay.
If you have smoking, it is already reduced, your efficiency is reduced, the area is gone. But you can increase the efficiency only by.
But is that proven? Like do with purpose?
I read a lot of papers. I tried.
Are you trying to scare me?
No. No.
No, actually, like during COVID times itself, simultaneously the daily air pollution was going on.
Actually, even
So
before COVID, so the group that I was part of at UNC, they were. So even when I was at UNC, there was a group doing tobacco smoke inhalation studies, especially because that was when 2016, 17, right? So the e-cigarette concept was coming up. Now they know that e-cigarette is also bad, but they were doing a lot of studies back then. And yes, so this kind of, as you said, like it doesn't matter what the particle is, there is size demarcation. One thing that we need to be careful while modeling smoking is you don't need to worry about the nasal cavity details, right? So the mouth gives a more direct access. So that means the 20 micron particles that might have been screened in the nasal cavity are probably going further downrange. So that is concerning in that sense. But the secondary thing that comes up, and I don't know how much we will explore that, is we have an inner replenishment going on. So the mucociliary fluids they recirculate, and that can clear out some of the pathogens, some of the contaminants. Now, whether that is sufficient for someone who has been smoking for the last 20 years, 30 years, that's a good question.
But I was on the end of trying to scale it. I remember. Trying to scale it.
And did he stop?
I don't think. But he stopped talking to me.
The other thing is like many smokers, since we are talking about smoking here, they also exhale through, they take in through the mouth, they exhale through the nose. That itself is a very interesting mechanics question. Because the flux going in through the mouth, and we try to not get things into the lungs, and we are exhaling through the nose. But still, it's an uncontrolled mechanism, right? There could still be some population going on down there. And I'm pretty sure no one has looked at it yet, but something that could be very interesting. Any other questions? Okay, so now we are again going back to sort of where we left off yesterday. But unlike yesterday we were just looking at single aerosol motion through fluid. I did not bring in anything related to the nasal cavity deposition or anything like that. But now we have to think about how we can connect the mathematics to the physiological system that we are trying to study. So this is now aerosol deposition. in the nasal cavity. So from yesterday, we are going to start using this equation that we had derived yesterday. So Stokes number dv bar prime dt prime is equal to v settling over the characteristic speed of the ambient flow times the non-dimensionalized g minus v bar prime relative. So just for the sake of completion of the lecture notes, let me write down these definitions. So v bar prime is v, which is the particle velocity, over the characteristic speed. V bar relative is v bar relative prime is v bar relative over u naught, or v bar minus v fluid, which is air over here, divided by u naught. So many times when we run these modeling studies and the papers that we've come across, they ignore the gravity. They don't consider gravity while trying to model these studies, including the paper that we saw last night. Everyone here tried to read the paper last night? So their sign had ignored gravity as well. So what is the argument towards ignoring gravity? That was a question that came up when I first started working on this field as well. Well, for the bigger particles, gravity does play a role, right? Like if you have 100, 150 micron particles, gravity would play a role. But the paper that you had read last night was it on drug delivery or something else? That was on deposition of what? Aerosols. What kind of aerosols? For pathogens, for infection transmission. So I think he first did a spatial modeling, like in stress, and then he tried to look at the health transmission. So he is talking about aerosols that are tiny. Aerosols are droplets that are tiny in the sense that anything bigger than 150 microns would a sediment of the gravity in stress. We are not going to inhale those things. We are inhaling just those very small particles. And for them, gravity could be ignored. Even if you do turn on gravity, you will see that the regional deposition does not change much because that inertial nature is not there. So since we are trying to develop an analytical framework for this, it's not a numerical framework yet, let's ignore that. That's because the particulates are small enough. So ignoring gravity, we are left with Stokes number dv bar prime dt prime is equal to minus v bar prime relative. Okay, let's now try to apply this. We are trying to get a physical derivation or physical understanding of what is going on in real scale. So let's now get rid of the non-dimensionalization and put in the terms over here. So Stokes number we derived yesterday is u naught rho p d square cc over 18 mu d. U naught is the characteristic speed, d is the characteristic dimension where we are studying the transport process, times d, v bar prime is v bar over u naught, dt prime is t over the characteristic time is derived from the d and the u naught that we have assumed. Right? And this is equal to minus v bar minus v fluid bar divided by u naught. Now let's take the denominators out from the differential parts. So we have u naught rho p d square cc over 18 mu d times 1 over u naught by 1 by d over u naught times dv bar dt. So I'm taking the cofactors within the differentials out, and this is equal to same as before. So minus v bar fluid by u naught. Let's keep on simplifying. So u naught rho p d square cc over 18 mu d. This u naught, so this becomes d over u naught square, right? Yeah. Correct me if I'm wrong anywhere. So dv bar dt is equal to same as before. So minus v bar fluid by u naught. Let's now see what we can cancel. We can cancel the d's. We can cancel this u naught with this and the other u naught as well. So we are left with rho p d square cc by 18 mu dv bar dt is equal to, let's take the minus sign out, so v bar fluid minus v. Okay, so let's box this up, and I'm going to call this as equation A. Let's get rid of the numbers from yesterday because things were already getting confusing. Now over here, what is this part? So if we check the dimension here, and personally I'm bad at remembering formulations, so I'll check the dimension what this implies. So rho p is L to the power of minus 3, d square is L squared, slip correction factor is dimensionless, divided by mu is kg per meter per second. So kg this. So this becomes 1 over t, right? Or rather, sorry, so this becomes 1 over t inverse. So this is t. So this is what we have derived as the particle relaxation constant, the tau. So this coefficient over here is a characteristic time so from the formalism derived yesterday, this is tau, which is the particle relaxation time. Okay, so let's now write this formula hence as tau dv bar dt is equal to v fluid minus v bar. Let's call this equation B. So again, V fluid is the inhaled flow velocity, V is the partial velocity, dV is the time rate of change, so that's the downstream flow. So now we are going to use this. So what we are trying to get at is, what if we apply this formalism into a real respiratory tract, or at least an idealized geometry that is inspired from a respiratory tract. So let's look at what I mean by that kind of idealization. So let's now consider an idealized nasal cavity. So when I say an idealized nasal cavity, I'm getting inspired by something like this. So we can think of this as sort of like a circular arc which eventually straightens out. We are not going to think about the lower airway or cognitive position line. We are just trying to develop a clean model for what is going on in the nasal cavity in the central space. So based on this, what I'm trying to draw here is something like this. And the cross section is consistent, and it could be the mid cross section from a real anatomy. And that's why I asked you guys to bring your MATLAB today, because hopefully we can solve some numerical examples by imposing the real cross section of that. So this is what I mean, but let me add the details here. So this is the central axis. Now, I already did a very bad job of sketching this. This is a circle, and this is an outer circle with same curvature. Let's assume that the center is here. Oh. Now, the distance to the central axis of the airspace is L0, let's assume, with the radius, the so-called radius being L. Okay. Now we are going to keep count of how far into the airway we are in using that S coordinate, or the value, let's not call it S coordinate because we are using a different coordinate system here, whatever the value of that S is. And we are going to track particles from here to where it goes. So there is the ambient airflux going through, but based on whether it is an inertially dominated particle trajectory, it can end up over here, right? There is nothing stopping. If the particle velocity is initially quite high, it just shoots and end up over there. Similarly, if the particle velocity is slightly lower, it can try to follow the streamline for a while, but it can end up over here. And that's where I'm trying to get at here. Because this equation that we have just derived, if I can use this equation, integrate it, and figure out where the location of the particle is going to be after a certain while as we integrate over time. If we see that it is landing on any of those volumes, and if we can develop the mathematical condition for it, we will have a classical mechanics guided framework to predict whether the particle is going to end up closing on the airway walls or if they're going to go for the downwind into the lungs. Okay, so that's the motivation. Okay, so what else do we need here? So this is, we are going to use the cylindrical polar coordinate system. So while it is a 3D model, but we are not assuming any kind of effect in the direction normal to the plane of the screen. We have this distance or the location as R. So wherever the particle might end up as it moves through this space, we can noted by r. Along with, we need another second coordinate, which is this theta. So knowing r and theta should be sufficient to know where the particle is. And what else do we need? I think that's it. Any question before we move any further? Do you think this is a self-sufficient setup to know about the particle dynamics, as well as we can use that equation that we have had before? Huh? Yeah, of course, we need the initial conditions to solve the differential equations, but I'm talking about the geometry, realized geometry that we have. The total length, that is relevant. At this point, we are assuming that this is a 270 length. So that itself will tell you what the total length is. And in fact, to develop the analytics or the analytical formula, we don't need the actual value of it. We're just assuming some symbols, right? But we are going to solve some real examples where we are going to use those specific numbers. So S is there, L0, which is the, let's use capital L. So this is something that we are going to need. Yeah. So I think we have all that we need for the simplest possible, yet something that captures all the details, so that we can use that differential equation that we have set up. So now we can write so what is going on here is a combination effect of the fluid velocity, the fluid moving, and the particles transporting, right? So let's first think about the fluid velocity. So the V fluid, can we express the V fluid in terms of the theta coordinate system? So what do you think should you write if you are trying to express V fluid in terms of the r theta coordinate system? True, but that's saying if you are, these are the two components, V R and V, right? Can you enforce more conditions in there, because we are trying to get something which is simplest possible. Yet captures most of the realistic information. Can we imagine that this is something? So if you think about a packet of air moving through the system, it is moving like this. I don't know if it would be clear enough, but this packet of air moves directly like this. So like this. So what that implies is V theta is always there, but can, if I say something like this, can you say something about what the VR is? If it's going straight along the angle. Exactly. So that is a reasonable estimate, reasonable, I would say, assumption that VR is zero. The flux is moving directly like this. So we can write the V fluid as some scalar number V fluid times theta unit vector. Similarly, not similarly, but of course, like if you resolve the components of the other phase that we are talking about, is the V particle. So V, so this is for the particle, this is for air. So V, now tell me what the resolution in these two components should be for the particle velocity. There will be V theta and V R, but can we say something about either theta or V? We cannot. We cannot. So it depends on the particle size whether it's going to follow the same lines or if they will fall out of the same lines. We need to chart out all the details. We need both the components for the particle velocity. So it should be V theta unit vector plus VR unit vector. Okay. So now let's. Are we stopping at noon? No.
You can just complete these things.
Okay, it will take a while to complete this derivation, but let me go somewhere and then continue. So these are the two representations for the two velocities that we are trying to think about. Now let's come to the equation that we have just derived. Tau dv bar dt is v fluid minus v, right? From the previous equation. Let's now plug in what we just wrote here. So tau ddt of v theta unit vector plus vr unit vector is equal to v fluid bar is v fluid theta minus v theta unit vector minus plus vr unit vector. Do correct me again if I make a mistake. So this is it, and let's now break the left hand side apart. So tau dv theta dt times unit vector plus tau v theta d theta of the unit vector dt of the unit vector plus tau dvr dt times the unit vector r plus tau vr dt of the unit vector r, right? I could have taken tau out, so let's take that. Okay, so now on this side, if I on the right hand side, if I club the theta and the r terms together, so v fluid minus v theta minus vr r. Okay, so the obvious direction where we are heading into is we will compare the theta and r components on the left hand side. That's the generic technique, and then they will still be differential equations which we have to solve using initial conditions. Fine. We have the terms for the theta and r components over here and over here. What is the derivative of the unit vector? What is dr dt and d theta hat dt in the cylindrical polar coordinates? Anyone remember this off the top of that? Probably not. I did not either. So if you remember, do they have access to the cloud portal now? No. I don't want them to access them right now, but they have access.
I said this is right.
Okay, so let me just quickly show you something here. So this is a screenshot that I took yesterday. Of course, I don't have internet either. But if you go to the lecture, I think you would find them under journal papers. So if you go to the journal papers, there is a document that I have prepared called coordinates. So there all the derivations for whatever the sub derivations which are not very, they're not very mechanics derivations, they're mathematical derivations for the coordinate system transformations, they're there. So if you have any doubts, you can go directly to that folder. Okay, let's carry on and write this one out. So you will see from that document in cylindrical polar coordinates, the time derivative of the unit vectors so see, I think it's called coordinates PDF on shared folder. So dr dt is theta hat, or the theta unit vector, d theta dt, and d theta dt is minus r hat theta dot. So now it becomes easy. We can plug in this into the previous formalism and then try to separate the theta terms and the r terms out. And that will help us form some revised version of the differential equations that we can solve, and then we'll see what comes after that. So let's stop over here, because then, yes. Theta hat. Dr dt. Yeah, it should be theta dot for both. It should be theta dot for both. But once you guys have internet access, you can quickly check if I typed it wrong. And in fact, when you guys have internet access, could you please take a screenshot of this page and bring it back on the next session? Okay, so let's stop for now.
Be that 2 again.
Yes.
This was 30.
Caveat.